Welcome to our lecture on nucleic acids. This is under the subheading Organic Macromolecules and in the topic of the chemistry of microbiology as part 8. This is forming our lecture 26 and the last lecture in this topic. The lecture is presented by us, Microbiology Biological Sciences. Kindly subscribe to our channel to stay tuned. You may kindly as well contact us for any queries or consultations through the number or email stated below. Nucleic acids. Nucleic acids include deoxyribonucleic acid and ribonucleic acids. They are named nucleic acids because they were first discovered in the nuclei of cells. The structural units are made up of nucleotides. The nucleotides are made up of three parts: a nitrogen-containing base, a pentose sugar, either deoxyribose or ribose sugar, a phosphate group that is phosphoric acid. The nitrogen-containing base are cyclic molecules composed of carbon, hydrogen, oxygen, and nitrogen atoms, and they are named adenine, thymine, cytosine, guanine, and uracil. Adenine and uracil are double-ring compounds referred to as purines, while thymine, cytosine, and uracil are single-ring compounds referred to as pyrimidine. DNA is composed of adenine, guanine, cytosine, and thymine, while RNA is composed of adenine, guanine, cytosine, and uracil. A nucleic acid that is lacking a phosphate group that is made up of only nitrogenous base and a pentose sugar is called nucleoside. Functions of nucleic acids. 1. The function is storage of energy. The nucleotide adenosine triphosphate, that is ATP, is the main energy in cells since it stores chemical energy in the form that can be utilized by cells. The bonds between phosphate, that high energy bonds in ATP, contain more energy than most covalent bonds. That is, more energy is released when they are broken down. Storage of information that directs protein synthesis, and they also function in the transfer of hereditary information. Nucleic acids are composed of long polymers of nucleotides, polynucleotides, that is, which are linked by covalent bonds between the phosphate of one nucleotide and the sugar of the next. The atoms of the bases of the nucleotides are arranged in a manner that hydrogen bonds readily forms between specific bases of two adjacent nucleic acids. 3 hydrogen bonds are formed between adjacent pairs composed of cytosine and guanine, whereas 2 hydrogen bonds form between an adjacent pair composed of adenine and thymine in DNA, or between adjacent pairs composed of adenine and uracil in RNA. Generally, DNA molecules are composed of 2 complementary strands. For example, if one strand has the sequence adenine, thymine, guanine, cytosine, and thymine, then its complementary strand has the sequence thymine, adenine, cytosine, guanine, and adenine. The two strands run in the opposite directions, that is, antiparallel. One runs from the 3s strand end to the 5 end, whereas its complement runs from the 5 strand to the 3s strand. The several hydrogen bonds between the strands stabilize the double-stranded DNA molecule structure at normal temperatures. The hydrogen bonds also twist the DNA backbone into a helix structure. Helix, thus the general structure of DNA molecule is a double helix with a few exceptions like the parvovirus that uses single-stranded DNA. Here is a structure of our DNA where we are having adenine. Adenine is composed of a phosphate sugar, a sugar, and adenine, which joins forms a covalent bond with thymine, and thymine nucleotide has thymine, sugar, and a phosphate. On the other side, it's linked with the phosphate group with the guanine, and cytosine thymine with the phosphate group. The double helix structure is as shown, one running from the 3s and the other one running from the 5s strand, where adenine and thymine are joined by double hydrogen bonds, while cytosine and guanine are joined by triple hydrogen bonds, and then they are covered with the sugar phosphate backbones. Differences between DNA and RNA. DNA are generally double-stranded, while RNA are primarily single-stranded. The pentose sugar in the RNA is ribose, while has one more oxygen atom than deoxyribose in DNA. One of the RNA's bases is uracil instead of thymine in DNA, and there are three major kinds of RNA. You have the messenger RNA for transfer of information, you have the ribosomal RNA, and you have the transfer RNA. That's what we call the tRNA. Thank you very much for viewing. We urge you to kindly subscribe to our channel to stay tuned. This lecture is presented by us, and feel free to contact us with the contact listed on the first page. Thank you.